SMOA stands for single molecule arrays, which is a technology that my lab developed approximately 10 years ago. The idea is to capture molecules, whether they're proteins or nucleic acids, into very tiny microwell arrays. The idea involves taking beads, tiny microspheres, we call them beads, and capturing on those beads an individual molecule of a protein or a nucleic acid by binding it to the surface using a selective binding agent. And so if we have now captured a single molecule of protein or nucleic acid, that molecule is labeled with an enzyme. The enzyme catalyzes many molecules of formation of fluorescent product, and that fluorescent product is really easy to see in those wells. So we look at an array of these, and where there is a bead with a molecule attached to it, it's very bright fluorescent. Those wells that have a bead where there's no molecule attached to it are dark, and we just measure the ratio of the beads that are fluorescent to the ratio of the beads that are dark, and that gives us the concentration. The goal of this is really to take individual cells, many individual cells, and analyze them at the level of being able to count how many molecules of a particular protein or nucleic acid exist in these individual cells. And what we see, and this has been demonstrated for other things, but not quite with the sensitivity that we have, that there is a tremendous amount of heterogeneity, that is variation from cell to cell. And the reason that we're interested in this, the ultimate application of this would be, for example, to take a tumor biopsy where you have maybe a few thousand cells or a few tens of thousands of cells, and to be able to resolve that biopsy in terms of looking at individual cells in that population as opposed to looking at the entire population, which is the way things are done today. If there's a rare cell that might in fact be the one that eventually kills you as a result of being able to escape a regimen of chemotherapy for cancer, we hope that one day we'll be able to analyze those biopsy samples with the type of resolution that would be necessary to be able to perform that kind of high-resolution diagnostic that then is predictive of what the outcome would be. This is really the premier meeting for analytical chemistry in both the ability to have a wide spectrum of technologies being presented, but I also think that people who aren't even analytical chemists come here to present their work on how new technologies such as nanotechnology can really begin to have an effect on being able to make ultra-sensitive measurements or measurements using imaging technologies. That combined with the exhibition really makes this a premier conference and meeting.